Hello and welcome to another of my eBiology Teacher podcasts. I'm Mr. Doc, and today we are going to be talking about DNA. This is the first podcast in our series on cellular reproduction, that is how cells reproduce themselves. And to understand that, we got to understand our pal, DNA, that guy right down there. See him? Hello. We're going to talk about how DNA was sort of discovered. We're going to talk about its structure and, of course, its function, because in biology, structure and function always go hand in hand. We're going to describe the structure and function of all three types of RNA. We're going to talk about complementary base pairing, which is actually the key, and then we're going to compare and contrast both DNA and RNA. So let's go ahead and get started. In about 1928, a guy named Frederick Griffith discovered that cells could transfer genetic information between each other. This means that cells had a way of communicating, and we've never heard them talk, so there had to be something that they used to communicate between each other. In the 1940s, a decade or so later, Oswald Avery discovered that cells missing RNA and protein could still transfer genetic information, but if they did not have DNA, that information could not be transferred, which indicated to us, and of course to Martha Chase and to Alfred Hershey in 1952, that DNA had to be the molecule that was responsible for heredity, meaning that DNA was the molecule that said, okay, here is the information you need to survive and to live and to grow and do all that things. DNA is the blueprint. And as I've already said, structure and function go hand in hand. They've got to, okay? So what are we looking at? So what, DNA, whatever, right? No, the next step is to say, okay, if DNA is so important, the first thing we need to understand is not what it does, but what it looks like. What's its structure? Because if we know its structure, then that will tell us a whole lot about how it functions. So in 1950s, that era, a guy named James Watson and another guy named Francis Crick were trying to understand how DNA worked. And to do that, they wanted to understand what it looked like. And one of their colleagues by the name of Rosalind Franklin was able to take an X-ray diffraction photograph of a DNA molecule. And what she looked, if you look down here, looked a lot like this. Okay, they kind of could see that the DNA molecule had these rungs and it had these spirals that went down both sides, and that these vertices or these rungs kind of kept it all together. They really didn't get a pretty picture like this, but what they did see was enough to come up with this structure, which they called a double helix. Now, the double helix is what won them a Nobel Prize. By the way, Rosalind Franklin didn't get hers. It's a scientific slap in the face, if you ask me, but it's just one of those things that happens that in history we're not that proud of. But anyway, they won a Nobel Prize because in discovering this shape of the double helix, which you see right here, okay, they were able to tell us a whole lot about our history and about how people work and how organisms in general work, because all organisms have DNA. So let's take a look at DNA. Well, what does it all mean? First off, DNA, or deoxyribonucleic acid, is composed of small units or monomers called nucleotides. Now, if you look on the screen over there, that's a picture of a single nucleotide. If you look at the diagram down here, what you're looking at is this is a single nucleotide right there, okay? So let's go through and look at the different parts. First off, a nucleotide is composed of a deoxyribose sugar. And I'm going to go ahead and correct something on the screen. If you look here, this shows that a deoxyribose sugar is 5-sided. That's wrong. It's actually a 6-sided sugar, so it looks more like a 6-sided diamond and less like a pentagon, okay? It's actually a hexagon, for those of you who are in geometry. All right, so it's made up of a deoxyribose sugar, a phosphate group, and then one of 4 nitrogenous bases, and those are what we're going to get into next. But if you look at this one on my DNA model, I have a deoxyribose sugar here where it says D. The phosphate group is here. Actually, on here it would be there. And then we have our nitrogenous base, which is denoted by the letter A, and it's the red part. This one's probably a little easier to see because the colors are more different. We have the deoxyribose sugar, the phosphate group here, and then we have the nitrogenous base here. Okay? Now, the nitrogenous bases are really where the important part of this whole understanding comes from. You see, there are 4 types of nitrogenous bases. We have adenine and thymine, and we have cytosine and guanine. The nice part about this is that they only fit together in a complementary way, meaning that, notice that the shape of C and G are opposites. That means they will fit together. The same with A and T. Goodness gracious, I got to get this thing to work. Hold a second. There we go. And now A and T fit together in very much the same way. Okay? Now, notice that if I try to hook up a G with a T, for example, that is not going to work because their shapes are not complementary of each other. Only G's and C's will connect, and only A's and T's will connect, right? So always, that's the way it's going to look. Those are called complementary base pairs, all right, or complementary nitrogenous bases. The double helix is a result of a couple of things. First off, the deoxyribose sugars, which are out here on the outside, connect to each other with a phosphate group from the nucleotide below it. So the phosphate group reaches up and grabs onto the deoxyribose sugar and then reaches out to grab onto the nitrogenous base. The deoxyribose sugars and the phosphate groups create this strand around the outside, which is kind of like the uprights of a ladder, and then the nitrogenous bases make the rungs. The rungs fit together, and because they fit this way, they twist. Okay? Now, the rungs here, I should go here because you can see it here, are put together by strong covalent bonds. Covalent bonds hold much stronger because they're sharing electrons. But the rungs here are held together by weaker ionic bonds or hydrogen bonds. Okay? The hydrogen bonds break pretty easily. Gee, I wonder why. Well, remember that this is a code. Okay? This whole thing is a list of instructions for us for everything that our bodies do. And the code is actually inside here where these letters are. But right now they're unreadable because they're connected to each other and I can't get to them. And of course, if we were to break this, then they would be out of order. So I need to be able to keep these in order, but I need to be able to get to them. So the outside is very strongly bonded in forms of a covalent bond, but the inside breaks apart very easily. Look at that, right? So now what happens, not that easy, hush. All right. Now what happens is I'm able to expose the information in my DNA into what looks like a readable sentence. A sentence in letters that my body can decode. So I need to go T, C, A, G, A, C, T, G, A, G, T, G. My body can read that sentence, and it can use it to do some pretty cool things. As a matter of fact, it uses it to do everything. But keep in mind that for these to pair up, they've got to pair up in the right direction. So let's take a look at this. This is what's called a 5' to 3' direction. This one has to go in a right order in order to connect. Notice that if I go 5' to 3' here, that's not going to work. They're not going to work. So what has to happen is this strand, which is called the lagging strand, has to turn around and turn upside down in order for everything to fit together. And as they do that, we're going to see that the pieces come back together and form that double helix that we're so fond of. So now that we understand DNA, we need to understand RNA. RNA's main job, or DNA's job, is to store information. RNA's job is to use it to do something. There are 3 types of RNA. mRNA, which is the messenger RNA, it goes to DNA and gets the information. rRNA makes up ribosomes, where the proteins are made. And tRNA are kind of the worker bees of the RNA. They go out, they get the amino acids needed to build certain proteins, they bring them back, and they actually build the protein. So RNA is pretty spectacular stuff. How is it built? Well, that looks a whole lot like DNA, doesn't it? And it is, except that, first of all, we're dealing with a ribose sugar. This one is 5-sided, not 6-sided like deoxyribose. And the nitrogenous bases are almost all the same, except that there is no thymine. In the place of thymine, there's uracil. That's RNA. So let's go over the differences real quick so that we can distinguish between DNA and RNA. First off, DNA is a double helix. That means it makes that twisted shape that we saw earlier. But RNA is single-stranded. It looks something like this, right? And sometimes RNA can double back on itself and it can connect like this and make little rings and whatever, but for the most part, it's just a single strand. Another difference is that in DNA, we have thymine, but if this were actually RNA, this thymine would be uracil, okay? So we have adenine, thymine, cytosine, and guanine in DNA, but in RNA, we have adenine, uracil, cytosine, and guanine. Finally, the base pairs are pretty much the same, except where you would expect to see a thymine in RNA, you'll actually see a uracil. So DNA pairs up AT and CG, whereas RNA pairs up AU and CG. So the last thing we really want to talk about is what do they do? More specifically, what's going to go on. And this is going to kind of preview what we're going to be covering in this unit. DNA acts as the blueprint for everything in the body. DNA can also be copied to make more blueprints, which means that we can make more cells. That's called DNA replication, which is the next thing we'll talk about. mRNA can translate this information from DNA and take it out to the cell, while tRNA can transcribe it into protein, and that's protein synthesis. So that's the third thing we'll talk about in this unit. So let's take a look at our objectives. Now, as we look at the objectives, I'm going to try to put my DNA back together. All right, the first objective is recount a brief history of DNA. Well, pretty simple stuff. First off, we had Griffith, who told us that DNA was, or that cells could communicate to each other. And then we found out that cells that had DNA could transmit information, but cells that didn't have DNA couldn't. So that led us to the Hershey-Chase idea that DNA must be the thing that's kind of responsible for the whole, heredity thing. Then Watson and Crick and Franklin discovered the shape of DNA through a X-ray refraction photograph, and they called it a double helix. Everybody jumped for joy, and we said, yay, double helix. We understand now. The double helix, though, gave us a really good idea as to how DNA really worked. The structure of DNA is that of a double helix, but the double helix is made up of a nitrogenous base for each nucleotide, along with a deoxyribose sugar and a phosphate group. The phosphate groups and the deoxyribose sugars make up the strands, while the nitrogenous bases make up the rungs to our DNA ladder. The structure and function of RNA is very similar to that of DNA. However, instead of a deoxyribose sugar, we have a ribose sugar, and instead of having the nitrogenous base thymine, we have the nitrogenous base uracil. However, the types of RNA differ on their job. For example, mRNA carries the message. rRNA makes up ribosomes where proteins are generally made. And of course, tRNA is the worker bee. They're the ones who go get the amino acids and make everything sort of happen. And finally, complementary base pairs, we've already hit on, is the fact that adenine and thymine in DNA connect. Cytosine and guanine in DNA connect. But then again, in RNA, adenine and uracil connect, and cytosine and guanine still connect. I know I'm a little distracted. I apologize. But look at that. I've made DNA. Woohoo! All right. To finally contrast DNA and RNA. First of all, DNA is a double strand, double helix, and RNA is a single strand. And of course, uracil and thymine are not in the same, meaning that DNA has thymine, RNA has uracil, and that both thymine and uracil will combine with adenine depending on whether we're dealing with DNA or RNA. I hope that was not confusing. If it was, feel free to rewind this and go back, take a look later, and learn more about our friend DNA. It will tell you everything you need to know because indeed it is the instruction book on how you are put together. Next time you see me, we're going to be talking about how we take this big monster and make a copy of it. And unfortunately, it's not as easy as pressing 1 on the Xerox, okay? So I will see you next time for another one of my eBiology Teacher podcasts.